Welcome everyone. I'm sorry for the long hiatus between the last video and this one, but today I'm going to be diverging a little bit from extrachromosomal DNA and talking more about another interesting aspect of cancer biology, which is how neurons can actually drive the proliferation of cancer cells. In two cell lines particularly, which are small cell lung cancer and pancreatic ductal adenocarcinoma, both pretty bad cancers, but both very interesting to study in their own right. The first cancer I'll discuss is small cell lung cancer, which is relatively uncommon as far as lung cancer subtypes go, but it does exert a variety of interesting manifestations. On the left here, you can see that small cell lung cancer is associated with many paraneoplastic syndromes, and these are just syndromes that are tangentially associated with the tumor itself, whether due to immune dysregulation or other means. And characteristically, small cell lung cancer causes a variety of endocrine and neuroimmune derangements. For example, it causes Cushing's disease by overproducing ACTH, which is essentially a precursor to cortisol. It causes antidiuretic hormone overproduction, which can cause low sodium levels and increased fluid retention. And characteristically, it causes Lambert-Eaton myasthenic syndrome, which is due to autoantibodies that are produced against voltage-gated calcium channel, which is, of course, very important for muscular function. So ultimately, this is because small cell lung cancer is a neuroendocrine tumor. It presents in these sheets of small purple or blue cells, you can see on the bottom right here. And because these cells are so aggressive in nature, small cell lung cancer is characteristically associated with very rapidly recycling tumor cells, similar to what we see in lymphomas. This causes your immune system to kind of rev up against anything endocrine, which is why we see so many of these manifestations. Characteristically also, small cell lung cancer has the propensity to metastasize or spread into the brain. The top right here, you can see a CT scan, and in yellow, you can see circled a metastasis at the gray-white matter boundary. And characteristically, this is very difficult to treat in small cell lung cancer, and whenever a patient presents with evidence of this cancer in their lungs, they also tend to get MRI of the brain and sometimes even whole brain irradiation just to prevent metastases. So with that very important background on small cell lung cancer in mind, we can now discuss this very interesting study in 2025 by Venkatesh et al., which is basically examining the progression of small cell lung cancer and its association actually with neuron activity, specifically neuroelectrical activity. And just to set some ground rules for this study, small cell lung cancer can be characterized by three distinct immunophenotypes, which are, first of all, its neuroendocrine origin. And these have three components within them. So it has parasympathetic activity, which is characterized by vesicular acetylcholine receptor, or VCAT. Sympathetic activity, which is characterized by tyrosine hydroxylase, which is the enzyme that converts tyrosine into dopamine and then eventually norepinephrine and epinephrine. And then the sensory or neuronal component, which is characterized by myelin basic protein, which essentially is the white matter in your neuronal tracts. So these are the three expression profiles they use if you want to go ahead and interpret the study. But they started out by actually inducing the development of small cell lung cancer in mice by administering an adenoviral vector with RPR2 luciferase, and that was delivered intratracheally. Then they either left the mice alone to develop the cancer, or they cut their vagal nerve, which is a very important nerve for digestive function. It comes from your cranial nerve 10 tract, and so they cut that, and they wanted to examine what the effect of cutting the vagus nerve would be on cancer progression. And on the right here, you can interpret these findings on your own, but essentially they found that with vagotomy, or with resection of the vagal nerve, mice with small cell lung cancer actually had less exponential tumor growth. They had less mean liver metastases. And so essentially it seems that the cancer is very responsive to neuronal stimulation in that regard. But of course they had to clarify further. So at the microhistological level, they actually found that malignant tumor cells, which were closer to axons within the intracranial space, and by close they had actually a definition of less than 100 micron distance, they actually had a higher Ki-67 score. And For those of you who don't know, Ki67 is basically a proxy for how proliferative a cell line is. So that was very interesting. And they also found that when cortical mouse neurons, which were a mix of glutamate expressing and GABA expressing neurons, when they were co-cultured with human small cell lung cancer cells, there was an increase in proliferation. However, very interestingly, this effect was entirely abolished by a specific voltage-gated sodium channel inhibitor called tetrodotoxin, which we'll go into a little bit later, but this essentially abolishes neuron activity because remember that sodium flux is very important for neuronal signals to function. So both of these studies also reinforce the previous finding that nervous system activity, particularly proximity to nerves as well as actual electrical function, is crucial for the progression of some of these cell lines. So just to go further into tetrodotoxin, which as I mentioned is a voltage-gated sodium channel blocker, this is actually a very powerful venomous agent. It's actually featured in Japanese cuisine. Very trained chefs actually have to be able to take out the poisonous sac in order to even eat this pufferfish delicacy because it contains such a potent amount of tetrodotoxin. But anyways, they found that with ablation of neuronal synapse activity, malignant cells actually lost synapse-associated genetic signatures. So when they used tetrodotoxin to try to limit these cells, they actually found that they could reprogram the small cell lung cancer to lack a sort of pro-neuronal malignant phenotype. And so that's very interesting that neuronal activity can actually influence the genetic signatures that are expressed by these cancers. So then this led on to their electrophysiological studies. And these found that on histology or microscopy, that for every 10 small cell lung cancer cells, there were 3 to 5 synapses, aka junctions between neurons or junctions between neurologically active cells, which is quite high for a non-neuronal phenotype cell line. And actually, 22% of these cancer cells produce their own spontaneous currents. You can see in figure D on the right here that in cells cultured with just CSF, and these are cancer cells by the way, they had these kind of oscillating, kind of turbulent graphs that are representing actually spontaneous discharge of electrical activity. And this was completely abolished with TTX or tetrodotoxin. And this is actually abrogated also by NBQX, which is a glutamate inhibitor, and gabazine, which is a GABA inhibitor. So all of these neurotransmitters are actually involved in the electrical activity that these cells were spontaneously discharging. And the rest of this figure actually kind of demonstrates the electrical activity of these cells and the proliferation index effect when you actually introduce some of these neurotransmitter blockers, suggesting that proliferation was actually a key derivative of these cells having these neuronal synapses. So next they did a series of optogenetic studies. And optogenetics is a fascinating field. It was actually recently invented by Carl Dieserath at Stanford University. And in optogenetics, what you do is that you, into your neurons of interest, you introduce a specific protein called channel rhodopsin. Channel rhodopsin is selectively activated by blue light. And so when it's activated by blue light, or when you expose these neurons to blue light, they start firing spontaneous electrical activity in response to blue light exposure. And so in this study line, what they did is that they specifically introduced channel rhodopsin into certain segments of the brain, such as the pyramidal neurons of the brain, and they co-cultured small cell lung cancer with them to see if blue light exposure via the proxy of neuronal activity with these neurons transfected with channel rhodopsin, whether that would actually, again, proliferate small cell lung cancer or promote the proliferation. And that's indeed what they saw. As you can see in all these studies here, there is always a statistically significant increase in proliferation and with tumor spread in these neurons stimulated with blue light via optogenetics. And so this is a very interesting takeaway. And again, it reinforces the point that neuronal activity seems to be directly related to the progression of these small cell lung cancer cells, which are uniquely responsive to neuronal activity. Interestingly, the majority of the signaling, again, was mediated by the neurotransmitter GABA, or gamma-aminobutyric acid, and it was depolarizing in nature, which means it was more stimulatory than inhibitory. Now, this leads on to the final wing of the study, And this wing of the study explored an interesting phenomenon of brain cancers and brain tumors in general, which is that brain tumors tend to present with seizure activity. In fact, this is one of the common presenting signs of a brain tumor, when you have a localized or focal seizure stemming from the tumor itself. And this is likely thought to be due to irritation of local nerves and such. And so the question they were asking with this study wing is that, is there a reciprocal neuron effect by small cell lung cancer cells? This is the first question they asked, because we know that obviously neuronal activity seems to be driving cancer progression, but does cancer progression also cause spontaneous seizure-like activity within these neurons? And they eventually found that there was indeed a microscopic co-localization of synapses, of both presynaptic and postsynaptic structures within these neuron tumor cell complexes, which is very interesting, that these tumor cells were also creating their own connections, independent connections with the neuron. So that was very interesting. They also found that these tumor cells seem to have increased kind of voltage activity, and that was causing kind of spontaneous neuronal discharge. And then the second question they asked is whether an anti-seizure medication like levetiracetam, which is very commonly used in controlling seizure activity from brain tumors, whether that could actually reduce tumor burden and proliferation, similar to what we see with tetrodotoxin and NBQX, which are, again, those neurotransmitter inhibitors. And they indeed found that levetiracetam, or Keppra, reduced tumor burden and proliferation. So, again, this suggests that not only is levetiracetam controlling seizures, but it's also proactively reducing brain cancer proliferation and growth, which is a very interesting takeaway, that levetiracetam could also be having a curative effect, which suggests that it should continue to be used even prophylactically for small cell lung cancer treatment and management. So that was that line of study. So the next cancer cell line that may be responsive to neurons is actually pancreatic ductal adenocarcinoma. Now, pancreatic cancer is one of the most unfortunate cancer types that we have to deal with. It is very fatal and incurable, usually progresses to death quite rapidly. And it is also very painful because pancreatic ductal adenocarcinoma tends to invade the neurons, and it causes a characteristic gnawing epigastric pain, which is very severe within this cancer subtype. And on the left here, you can see a diagram of that process occurring, especially invasion into the neural space. And on the right here, you can see a CT of the abdomen, which shows something called double duct syndrome. And this is when a pancreatic tumor, particularly in the head of the pancreas, causes dilation and obstruction of both the pancreatic duct, which of course contains pancreatic enzymes, and that itself kind of wears away at the pancreas, causing autodigestion, as well as obstruction of the bile duct coming from the gallbladder and the liver, which causes jaundice and hyperbilirubinemia, so pretty severe manifestation. And in this study about pancreatic ductal adenocarcinoma, the major takeaway they gleaned from this study is that although neuronal activity itself does not by itself promote pancreatic ductal adenocarcinoma growth or change in phenotype, actually there is a neurotransmitter called glutamate that, independent of electrical activity, tends to stimulate and tends to change the phenotype of pancreatic ductal adenocarcinoma. And this is because pancreatic cancer can express a protein called NDMA receptor. And this is activated by glutamate, that neurotransmitter that we discussed earlier. And this causes the expression of calcium channel-dependent proteins and causes the formation of pseudosynapses, which may kind of feed into the growth of these pancreatic ductal adenocarcinomas. You can see this process occurring here, the diagram in the bottom right. So it's interesting that this is not happening due to electrical activity, but this is happening due to neurotransmitter expression. And this is a very interesting insight into this non-neural cancer. So I hope you learned something interesting from this study, and I think this is a very rapidly growing field of research now that we, are targeting more brain metastases, which are a huge, kind of confounding factor in the field of oncology because they're very difficult to treat outside of basically neurosurgery, which is not a great option. And so my sources are listed here. The first study is by Venkatesh et al. And the second study is by Demir et al., both published in 2025. So this is a hot topic of research. So thank you.